Hello folks, in this video we're going to take a look at important nucleotide containing compounds involved in metabolic pathways. I'm kind of amazed I was able to get all of those words out in one sentence. Now the first molecule we're going to take a look at is ATP, and we took a look at ATP when we were looking at esters. So this may be a little bit of a review of sorts. Now this right here is an ATP molecule, and it has different parts involved in it. And so we're going to need to know the different parts, and we're going to need to know how they interact with each other. So to get us started, this two-ring structure right here, this 6-member ring and this 5-member ring right here, this right here is called adenine. Adenine. And then this right here, this looks like a carbohydrate, and it is. This right here is ribose. So ribose has that OSE ending on here that tells us it's a carbohydrate. And then the adenine and the ribose, if we combine these two together, adenine and the ribose together, this gives us adenosine. Adenosine. Okay, and that is where we get the term ATP from, is adenosine triphosphate. 1, 2, 3 different phosphate groups. And a little bit about what's going on here with these phosphate groups is we have two different types of bonds. Right here, this bond here is different than the bond here and the bond here. And what we have is we have a carbon, an oxygen, and then a phosphate double bond O here. And so this one right here is called a phosphoester bond. So phosphoester bond. Phosphoester bond. And that's different than the bonds here. Over here, we have a phosphoanhydride bond. So phosphoanhydride bond. Okay, and there's two of them here. And phospho coming from the phosphate group, and then anhydride. We've seen anhydride before. This is removing water. And so when we say anhydride, it's missing water. But if we add water, we can break these phosphoanhydride bonds apart. So what I'm going to do here is I'm just going to say my ATP, here, ATP is all of these together. So it's adenosine triphosphate. And if I were to add water to this, HOH, add water, then what I'm going to do is I'm going to end up cleaving one of these phosphate groups. And so I'm going to start by cleaving this one, and then if I do it again, I would cleave this one here. So my first cleaving, my first water that I add here, I'm going to break this phosphate group off, and so that would break this bond here. So I'm just going to go like this. And then we end up with the oxygen like that. And then this phosphate, I'm going to redraw it down here. I'm going to go phosphate, okay, and then my oxygen, double bond O, and then O like this. And then what gets added right here is going to be the OH from the water. So I'm just going to take my OH from the water here. I'm going to go OH from my water, okay? And then I'm going to be left with a hydrogen ion. So on the other side here, I'm going to put my hydrogen ion, because that's left over. The OH went here. And then what is left? Well, I have and instead of being triphosphate, it becomes diphosphate, ADP, okay, adenosine diphosphate, plus energy. So energy is going to be released. So some small amount of energy that can be used for biochemical reactions powering the body somehow. Now, that's removing one phosphate group. Now, if I have what's left over here now is just two phosphates, so it's ADP. So over here, I'm just going to go, all right, well, let's start out with our ADP, ADP plus HOH, okay. I'm going to add another water, and then I will be left with a hydrogen ion, just like I was before, hydrogen ion, because I'm just cleaving this bond right here, just like I did before. And then I'm going to be left with adenosine monophosphate. Adenosine monophosphate, one phosphate is left, plus more energy. More energy like that. Okay. So some people ask, why don't we remove three phosphates? Like, we can remove two phosphates, but why not the third one? And the reason is this is a different type of bond here. This is a phosphoester bond, and this is a phosphoanhydride bond. And the enzyme that would catalyze this reaction here, this here would be a hydrolase enzyme, is going to be specific for this, okay? So adding water here to break these off, but it's not going to work for this, right? So the enzymes are specific, okay? So we can remove two phosphates, but we don't remove a third using the same enzyme. And every time we do this, we're releasing energy. So if we have ATP, we can release here two hydrogen atoms and some amounts of energy like that, ATP. This big, scary looking molecule here is flavin adenine dinucleotide. Okay, that's a big mouthful, lots of syllables there. And so what's going on here? Well, we have some things we should recognize. We have ADP here, so adenosine diphosphate. There's two phosphates here. So I'm just going to go like this and call this one ADP, adenosine diphosphate. And then over on this side here, this odd looking structure that looks kind of like a snowman. Do you want to build a snowman? So this snowman looking thing here, this is flavin. F-L-A-V-I-N. Okay, and then this in the red right here, this right here is called ribitol. And then ribitol and flavin, if we bring these two molecules together like this, both of those together, this is riboflavin. Riboflavin, and you might recognize riboflavin as a B vitamin. A B vitamin. All right, so how does flavin adenine dinucleotide, what FAD, okay, what's the deal with that name? So FAD, the flavin part of this comes from here, okay? So flavin, and then we have adenine, which is over here, and dinucleotide. This is a nucleotide, this over here is a nucleotide. So that's where the FAD gets its name from. Now, FAD is a cofactor for enzymes, so it helps enzymes to do certain things. And so what's going to happen here is we're going to take this molecule here and we're going to add some hydrogens to it. And so what can happen is we can break this bond here, we can break this bond here, and we can add a hydrogen to this nitrogen and this nitrogen here. Okay, so but before we do that, this version of the molecule without those hydrogens added like that, we would say that this is oxidized. This is the oxidized form. So I'm going to say here oxidized, like that. And we're going to add a couple of hydrogens. So add a hydrogen ion here. Well, we're going to add 2 of them. So let's see if I can count here. 2 hydrogen ions. And then just for our counting, we're going to say that we're going to add a couple of electrons with that too. A couple of electrons. Because what is a hydrogen ion? Well, it's just a hydrogen atom that's had an electron removed. Okay? And if we do that, then this reaction can go in both directions. We're going to end up with FADH2, 2 hydrogens, all right? And this version here, we would say is reduced. That's been reduced. And so what's going on with that? Okay, so we're going to add 2 hydrogens. So to add 2 hydrogens, we're going to take this molecule here, we're going to break this bond, and we'll add a hydrogen, like that. And then we'll take this bond here, and we'll add a hydrogen there. Okay, so if we do that, now it's been reduced. That is to say that we've added some hydrogen atoms to the molecule, and it goes from FAD to FAD2. All right, and you might be going, okay, why would we want to do that? What the heck's the point of all of that? Okay, so say you have a molecule that has a couple of carbon atoms like this, and a couple of hydrogens like that. Okay, and you wanted to remove those hydrogens and create a double bond here. Well, then you would add FAD, you could add FAD to it, okay, as a enzymatic cofactor. And what it would do is it would take those 2 hydrogens and add them here, right? So then this becomes, all right, we'll end up with FADH2 like this. And then we've removed these 2 hydrogens, and we'll end up with a carbon-carbon double bond like that. Okay, and so we end up going here from this molecule here is reduced. I'm going to go RED, reduced, and then this one over here becomes oxidized. Okay, and our FAD goes from being oxidized to being reduced. Okay, and all that is just fancy chemistry jargon to just say, well, we've just taken a couple of hydrogens here and brought them over there. Okay, and so that's the purpose here of FAD, is it helps us to remove hydrogens and create double bonds. This molecule here is called NAD+. Now the NAD stands for nicotinamide adenine dinucleotide. And that's a mouthful. It's a lot easier to just say NAD+. But just to kind of orient ourselves around this molecule, we may recognize this part right here as adenine diphosphate. Here's our adenine, and here's our 2 phosphates. So I'm just going to call this portion right here like this ADP, adenine diphosphate. And then over here we've got ourselves a ribose molecule. Ribose, just like we have down here. And then this portion up here, this right here is the interesting part here. This is the nicotinamide. So I'll just write this up here. Nicotinamide. And this is also a B vitamin. So B vitamin. Okay. And this is the part that we're interested in. This portion over here is just gobbledygook for right now, but here's a nitrogen that has 4 things coming off of it. Got a single bond, and a double bond like that. And because of that, it gives this nitrogen a positive charge to it. So all of this thing right here is got a positive charge. So we would call this here NAD+. So I'm just going to go like this and just call this here NAD plus. Okay, NAD plus. And what we're going to do is we're going to take this portion here and we're going to add some hydrogens and electrons to it. So if we take this and we add, say, 2 protons, okay, and 2 electrons, then what's going to happen? Well, we're going to end up then, we're going to break one of these double bonds here, and then we're going to add a hydrogen. And so what the product ends up looking like is all of this, and I'm not going to redraw all of this, but I'm just going to take this portion here, and I'm just going to redraw it down here, where I'm going to go like this, and I'm going to have the rest of the molecule, that's all of this, and then I'm going to have my nitrogen here, okay, and then we broke that double bond. We broke the double bond here, and we have 2 left, and so those move to there, and we have had a hydrogen here. It's not drawn, but we have a hydrogen there. And how do we know there's just one hydrogen? Well, we've got a double bond here, we've got a single bond here, so that carbon atom has 3 bonds, it needs 1 more, so there'd be a hydrogen. Okay, so I'm just going to put a hydrogen like that, and we're going to get a hydrogen from here. And so I'm going to attach that extra hydrogen here, and I'm going to do it in blue just so we can see where that came from. It came from up here, and we're going to add those 2 electrons. And by adding those 2 electrons then, we end up taking this from being positive like this, and it becomes neutral. Okay, and I should put my carbon double bond O and then amine group here like this. Okay, so this new molecule here is going to be NAD. Okay, so I'm going to call this NAD, and then because we added the hydrogen on there, it becomes NADH. And we end up with, we just added 1 hydrogen. That other hydrogen ends up being extra, and so I'm going to go like this, just to show that one is extra. So we start out with 2, 1 ends up here, the other one ends up over here. It's just left over, and it just kind of goes off into the tissues and such. Okay, and so really our equation here is NAD plus 2 hydrogens plus 2 electrons yields NADH plus a proton or a hydrogen ion like that. Okay, and maybe I should rewrite that and just kind of throw all that out there. We're going to do NAD plus 2 hydrogens, ions, okay, plus 2 electrons yields then NADH plus a hydrogen ion. And then this original form right here, this is our oxidized form. And then over here where we added the hydrogen, this is our reduced form. Okay, so why would you want to do any of this? Well, this NAD plus is a cofactor. And so it's involved in helping chemical reactions to happen, or helping our enzymes. And so if we, gosh, I'm running out of space here. I'm going to erase some stuff. I hope you don't mind. Is that okay? I'm going to just erase this part right here. And like this here, just giving us some space. And say, all right, well, what if we had a little more space? Okay. What if we had something like this, where we had carbon, OH, OH, I can spell OH like this, okay, hydrogen, and then R. Okay, so this right here is an alcohol, okay? And it's a secondary alcohol because it comes off of a secondary carbon like this. And we catalyze this or react this with NAD+. Okay, so NAD+ is going to pull 2 hydrogen ions off of this. And so we end up then with our alcohol losing 2 hydrogens. And so we end up with a ketone like this. A ketone because we removed a hydrogen, this bond goes up, and we end up with a ketone. And then over here, we end up with NADH plus a hydrogen ion, okay, plus a hydrogen ion like we did here. Okay, so this just helps us really to remove a couple of hydrogens like this and then create a ketone. So basically we're just removing hydrogens. All right, so that's just another one of these biomolecules involved in metabolism. You would not be expected to draw this. What you might be expected to do is say, all right, what's going to happen with this? If we add hydrogen, where's it going to go? Okay, and maybe can you recognize some of the different parts of this structure, such as the ADP, adenosine diphosphate, the ribose, and the nicotinamide. This big molecule here is called coenzyme A. And coenzyme A is sometimes abbreviated as CoASH. And we'll explain what all this is here. So this right here we may recognize as adenosine diphosphate. There's 2 phosphate groups here. But what makes it a little bit different is right here where we have this hydroxyl group is we are going to put in a phosphate group like this. So like one of these, phosphate group just substituted right here. So if we take this big molecule here and just go like this, we're going to go like that. Okay, so this is phosphorylated because it has the phosphate on here. So phosphorylated ADP, adenosine diphosphate. All right, and then this portion right here that's in the red, this here is pantothenic. This is pantothenic acid. Pantothenic acid. And this is another B vitamin. It's another B vitamin. And then over on the far end, over here, this group right here where we have the nitrogen, and then we've got, oh wait a minute, I think I'm missing a carbon here. Shame on me. Okay. I bet you were thinking, oh wait a minute, what's he doing there? This is wrong. Yep, this is, what am I doing? Okay, there we are. Shouldn't have that double bond oxygen on there. That was wrong, my bad. I apologize for that. Shame on me. Okay, so this part right here with the nitrogen, and then the SH right there, this is 2 amino ethane, because there's 2 carbons, thiol, because of the S there. T-H-I-O-L, thiol. All right. So those are the parts of this big, scary molecule. And what does it do? What's the purpose of all of this? Well, the purpose of coenzyme or CoA here, the coenzyme A, is to transfer acetyl groups. okay? And so I'm going to take this big, long molecule like this, okay? And I'm going to call this CoA, all right? So I'm just going to go and call this CoA, CoA. And then it has this SH here, okay? So I'm going to go S, and then instead of the H, if we attach an acetyl group to this, that would look like this, where we have the carbon, double bond oxygen, and then CH3, okay? And this acetyl group, I'll call this acetyl. This acetyl group, you might recognize that acetyl group that if we were to remove this here, okay, and we would have the carbon, double bond oxygen, CH3 like this. There's two carbons double bonded to an oxygen, okay, okay. And then if we were to put an OH on there, that's acetic acid, okay. So that's acetic acid. But if we just take that OH off there, then instead of acetic acid, it becomes an acetyl group, okay. And so coenzyme A's job is to carry acetyl groups and move it from one place to another and to catalyze reactions. So if we attach the acetyl group like this, then this has a new fancy name. So instead of being coenzyme A, it becomes acetyl CoA. Acetyl CoA. Acetyl coenzyme A. All right, and this is another molecule involved in biochemical energy production. Its job is to transfer acetyl groups, and it is derived from a B vitamin. Those are the things to remember about coenzyme A.